This tutorial walks through cross-species features in RGD's JBrowse 2, including variant tracks, ortholog tracks, and linear synteny views across rat, human, and mouse. To access JBrowse 2 from the RGD homepage, go to rgd.mcw.edu and select the JBrowse button under the Analysis and Visualization section. Or you can go to the Analysis and Visualization tab in the menu at the top of the page and select JBrowse Genome Browser from the drop-down menu. Both options will bring you to this page. Select Rat Assembly GRCR8 found under the JBrowse 2 header. Alternatively, JBrowse 2 can also be accessed from a gene page. We will use the rat gene adenylate cyclase 3, or ADCY3, in this example, so we type the gene into the search bar and press Enter or the magnifying glass icon. In the search results matrix for ADCY3, select the linked number under the rat gene, and select adenylate cyclase 3 from the list. This brings you to the rat gene page for ADCY3. Go to the Position section and select GRCR8 under Genome Browser's JBrowse. This takes you to a new JBrowse 2 session. First, let's adjust the window size. Here we see that accessing JBrowse from the gene page automatically opens to the position of the ADCY3 gene with the rat GRCR8 genes and transcripts tracked displayed, as well as the regions immediately up and downstream of ADCY3. There is also a menu to the right that shows the available tracks that can be selected and opened. To view more of the surrounding area, click the minus magnifying glass icon in the zoom slider or move the slider to the left. To zoom into a smaller region, move the slider to the right or click the plus zoom icon. Or you can click and drag in the detail position bar to select the region and choose Zoom to Region from the pop-up menu. I'm going to select the minus zoom icon to go back to our original view. In the track selector in the right sidebar, there are a number of tracks that could be of interest, including tracks that highlight genes, QTLs, and strains associated with major disease categories, gene models, genes that interact with chemicals in various categories, and tracks for QTLs and mapped strains, including congenic and mutant strains. Here, we will focus on the ortholog and variant tracks and the synteny data. To look at variants, click on the bar labeled Variants, and it will open to show subcategories for damaging variants, European Variation Archive, microsatellite markers, phenotypic alleles variants, and strain-specific variants. For this example, we will display the damaging variants in the WKY/N rat strain. To do so, click on Damaging Variants and scroll down to the track labeled WKY_N 2020_damaging. Click on the box in front of the name to turn the track on. Here is the damaging variant in the WKY/N rat strain. Clicking on the variant will provide featured details in the right sidebar. You can also search the RGD ID number on the RGD site for more information. Likewise, to view the variant associated with the ADCY3 EM2 MCWI allele and WKY-ADCY3 EM2 MCWI knockout strain, click Phenotypic Alleles Variants. Turn the rat GRCR8 RN8 Phenotypic Alleles Variants track on by checking the corresponding box. Again, if we click on the variant of interest, feature details will appear on the right. To help users see genes in their species of interest that have orthologs in other RGD species, RGD has created ortholog tracks for JBrowse 2. In the track selector, click the Orthologs category to open the track list. You will see that RGD provides ortholog tracks between genes in GRCR8 and at least one assembly for all of the other species in RGD. To view the orthologs in the most recent human and mouse assemblies, select GRCR8 rat-GRCH38.P14 human and GRCR8 rat-GRCM39 mouse. We need to scroll down to see both of the tracks we just selected. You can also adjust the window size, which we will do later. You will see that the tracks for human and mouse are different colors. The blue ortholog glyphs for human indicate that those genes are reversed compared to genes in rat. The orange glyphs for mouse indicate that mouse genes are in the same orientation as rat. To confirm that the region in human containing the ADCY3 gene is reversed compared to rat, go to the menu in the black bar at the top of the window and select Add, and select Linear Genome View. A sub-window will open inside the larger JBrowse 2 window. Select GRCH38 Human in the Assembly dropdown and type ADCY3 in the box next to it. Click Open. It is common for a gene to appear in multiple tracks, for instance, in the Ensemble Gene Features tracks, one or more of the disease-related tracks, or the NCBI Gene Features tracks. If this is the case, a pop-up will appear with the list of tracks that gene appears in, as is shown here. To display the human GRCH38.P14 HG38 genes and transcripts tract, select the Go button. Here I will adjust the window to see both tracks. And before we continue, I wanted to note that I unselected the damaging variants track by deselecting WKY_N and minimized the 3 other tracks we previously selected by adjusting the window size. Now we see the 2 sub-windows with the GRCR8 rat assembly open at the top and the GRCH38 human assembly open on the bottom. You can see from the arrows on the transcript icons that ADCY3 and the other genes in the vicinity are reversed in human compared to rat. The gene symbol for each gene is displayed below the transcript glyphs. If not all of the transcripts are being displayed and you are not able to see the gene symbol, you can easily tell what the gene is by mousing over the gene region. The gene will be highlighted in gray and a pop-up will display the gene symbol. One advantage of the parallel linear genome views is that all of the tracks for an assembly are available for all of the views displayed. Earlier, we had selected 2 ortholog tracks for human and mouse. Here, I removed the ortholog track for mouse. And now we see that the rat genome view shows the genes and transcripts tract, the phenotypic allele variants track, and the ortholog track between GRCR8 rat and GRCH38 human in the region around ADCY3 in the rat browser. Remember, the blue color indicates that the entire synteny block is reversed between rat and human. Click the block glyph to bring up the featured details display in the right sidebar. This display provides the positions and links of the corresponding genes in the 2 assemblies. The human genome view shows the genes and transcripts tracked. We can also add tracks by clicking on the hamburger menu, select Variants, select ClinVar, and select ClinVar-SNPs and indels pathogenic. Scroll down to see the track. This shows only the ClinVar variants designated as pathogenic or likely pathogenic, of which there are several that overlap the ADCY3 gene. This allows us to see what disease-associated variants exist in the human orthologous region. If we scroll over a bit, we also see variants in the upstream region containing the CENPO, LOC, and PTRHD1 genes. We can also select the rat-human synteny track from the Available Tracks menu. These blocks are generally large for closely related species, as we see here, where this block spans about 33 to 35 megabases, reflecting the large conserved syntenic region typical between rat and human. The synteny tracks in the linear genome view make it easy to see synteny information for 2 species or 2 assemblies. To explore synteny in more detail across more than 2 species, click Add in the black menu bar at the top of the window and select Linear Synteny View. A selection subwindow will open within the main JBrowse 2 display. When the selection pane first opens, both row 1 and row 2 will show the RAT GRCR8 assembly. Click the drop-down for row 2 and select GRCH38 Human. To add the most recent mouse assembly to the synteny view, click the blue button labeled Add Row. In the drop-down for row 3, select GRCM39 Mouse. Once assemblies have been selected, confirm the tracks to be displayed using the arrows to the right of the assembly selection boxes. When the first arrow is highlighted in gray, you can select and/or confirm the track for the rat-human comparison. In the list on the right, existing track should be selected. You can also select New Track and follow the prompts to upload your own data. But for this example, we will keep existing track. You can also click the box that lists the two assemblies and select the line with the same text in the drop-down list, but it is not required to move on to the next step. You do, however, have to click the lower arrow beside the assembly selection boxes. The line at the top right will say Synteny dataset to display between row 2 and 3, and should again have existing tracks selected. In the lower left corner of the pane, click Launch. In a new subwindow, the initial display in the linear synteny view shows red lines that link the synteny blocks between species across the entire genome. Chromosomes are listed across the top of each section of the display, with the width of each column determined by the relative length of the corresponding chromosome. At the top, there are three search/location boxes for the three assemblies displayed, shown in the same order as the display below for rat, human, and mouse. To zoom into the specific region of interest, search for the gene symbol ADCY3 in each box successively. Shown here for rat, when you type the symbol into the box, you will get an autocomplete dropdown. Select ADCY3 from the dropdown. As before, a selection pop-up will be shown that lists all of the tracks containing that gene. In this case, select the NCBI Genes Only track. Repeat this process and select ADCY3 for human and mouse, selecting the Genes Only track. This track shows a single bar glyph for each gene rather than the display of all of the alternative transcripts, consolidating these tracks. To view more of the regions surrounding the ADCY3 gene in the three species assemblies, zoom out using the -zoom icon at the right end of each genomic position bar. You can use a combination of the zoom out, zoom in, and selecting a region in the genomic position bar by clicking and dragging in the track to fine-tune the size and position of the region displayed for each species. Instead of navigating and adjusting each genome independently as we were doing, we can turn on the Link Views setting. When the setting is selected, panning and zooming in one genome view will automatically adjust the other view to maintain the correspondence shown by synteny lines. This may make it easier to explore syntenic regions interactively. Now if you select the zoom in icon, all of the tracks will adjust together instead of individually. You can go back and unlink the tracks to fine-tune your view if needed. Additional tracks can be added to any or all of the syntenic regions shown. Click on the hamburger menu and select Row Track Selectors. To add the phenotypic allele variance track for the rat display, choose Row 1 Track Selector GRCR8. The Track Selector menu is displayed in the right sidebar of the main JBrowse 2 window. As before, click the selection box for the rat GRCR8 phenotypic alleles variance. The variant associated with the WKY-ADCY3 EM3 MCWI mutant strain is shown, aligned with the ADCY3 gene. Follow the same procedure to see the Row 2 track selector for GRCH38.P14. In the track selector, select Variants, select ClinVar, and choose ClinVar-SNPs and indels pathogenic to view the variants in this region designated as being pathogenic or likely pathogenic for one or more conditions. I'm going to adjust the window and zoom out as we've done before. I am also going to adjust the opacity by using the slider at the top. The current view clearly shows the reversal of the human synteny block compared to rat and mouse, both by the gene order and by the distinctive X pattern of the lines in the synteny tracks that join the orthologs between the genome assembly tracks. In terms of gene order, the ADCY3 gene is outlined in each of the genome tracks, and for reference, the DNA JC27 gene, which lies immediately upstream of ADCY3, is starred, showing that it is chromosomally 5' of ADCY3 in rat and mouse, and chromosomally 3' in ADCY3 in human. Hovering over a synteny block will slightly darken it and display a box with details about the corresponding rat-human synteny region. If you click on the synteny track, synteny feature details will pop up on the right side panel, and we can do the same with the human-mouse tracks. We can now see more clearly the X pattern that shows the connections between the synteny tracks. For more information, visit the RGD help page, which can be accessed from the options at the top of the RGD homepage, or you can contact us by filling out the form on the contact page or sending us a message through this feature on the bottom right of the page, which appears on every page on the RGD site.